Let's go meet the rest of the gang.
Let us meet the students.
Hello everyone, my name is Nicole Mullen. I'm a second semester graduate student here at Fort Valley State University's Biotech program. And inside this specialty plant house, we have 3 different types of hydroponic systems that I'm going to show you today. So the first type is the NFT system. This system, all of our systems, by the way, they use a pump that helps to irrigate the systems or to irrigate the crops continuously. So specifically with the NFT system, it uses a rock wool which helps to absorb the nutrients so that the plant can get as much nutrients as it can. So what you do with the rock wool is you would open it up like so, and then you would put the plant inside, and then you would cover the rock wool back, and then you would place it back into the slot, and from there the rock wool will soak up all of the solution, and so the plant will get as much of the solution as possible. So the second system that we have is called the tower garden system. This system, it sits vertically and the pump sits at the bottom. And there are tubes that run all the way to the top, which allow for the water to recirculate back down to each individual crop. And what we have here in our tower today is butter crunch lettuce. This experiment has been going on for approximately 5 weeks today, and soon we will be harvesting. So now our third system is called a deep water culture. The deep water culture is just like the NFT system, but the main difference between the deep water culture and the NFT system is that the deep water culture allows for a bigger amount of solution to be exposed to the plant. So this is turmeric right here, and the way turmeric grows is it grows in the root or up under the ground. So the deep water culture system actually comes up to the root, or where the root and the stem actually separate, and that actually allows for more of the solution to actually get to the roots to, I guess, irrigate. So after that, I believe that we will show you the rest of the. Hey,
what you up to?
Nothing, girl. I know you was in here. Well, this is Elena Cheney, and she can show you the rest of the specialty plant house.
Hey guys, so my name is Elena. I'm a second year graduate student here at Fort Valley, and I'm from Augusta, Georgia. So here in our specialty plant house, we have several different plants that originate from all parts of the world. Each plant has its own unique use and value that we can appreciate. So here are some of our neem trees, also known as Indian lilac. Neem originates from the Indian subcontinent in Africa. So neem leaves can be eaten raw as a vegetable, and they can also be soaked in water, and then that brewed solution can be used as a natural insecticide. In cultures such as in India, they use it medicinally for different treatments for skin conditions, regulating blood sugar, such as in diabetes, and even to treat cavities. Over here in our NFT system that Nicole pointed out earlier, we also growing a plant called paper rallies. Now, paper rallies may be most commonly known for its natural treatment against the SARS virus, but in addition to its antiviral properties, it can also help to fight off bacteria. So the plant can be eaten raw, and when eaten raw, it can lower blood sugar. And we've also found some of its extracts in high-end face lotions and creams in countries such as Japan. So Papyrus is a really great plant. It's growing very well here. And so one thing about our NFT systems are the plants that we grow, because of the freshness and the rapidness that it can grow in this system, it can also be used as a business. So if you're interested in that, it would be a great subject to study on. So as we walk along throughout our greenhouse, you'll start to see over here to my right, we have some more of the tower gardens that Nicole was mentioning, growing different types of lettuce. We've got some butter lettuce, some romaine. We also have some red lettuce as well. We also have another deep water culture system growing some of our turmeric. We got some more paper rallies. Above me here, we have some different varieties of spinach. And then we also have a newly designed deep water culture system growing turmeric. Back here is our papaya tree. Now papaya originates from South America, and we mainly know it for its sweet fruit. But in some cultures, such as in Asia, they steam the leaves and actually eat it as spinach. Now papaya leaves have been used to treat malaria, and they've also been smoked to help relieve asthma. So I'm gonna walk you guys some more, and we do a lot of different projects here in our greenhouse. So you'll see some tools that we use to accomplish that. We have a saw over here. It just allows us to conveniently be able to cut wood at the measurements that we need to build different things like raised beds and platforms. And then we also have grow tables that allow us to keep our plants with time irrigation. So the last thing I want to point out to you is our moringa tree. So now moringa is a fast growing tree from India. It's mainly used to combat malnutrition, especially in children and nursing mothers. And that is because 100 grams of fresh moringa leaves has about as much vitamin C as an orange, 9.4 grams of protein, more iron than spinach, and 185 milligrams of calcium. So talk about a natural multivitamin. So I want to show you all back here where my other lab mate, Jeff, is waiting to share some sweet information with you.
Hi, my name is Jeffrey Lester, and I'm from Kennesaw, Georgia. I'm a second-year master's student here at Fort Valley State University, and here we are in the specialty plant house, Stevia Field. If you get on down here with me, you'll get an up-close shot of Stevia rebaudiana. Stevia rebaudiana is a perennial shrub that is native to Paraguay. It produces secondary metabolites that are 100 to 400 times sweeter than traditional table sugar. The growing season for Stevia rebaudiana is roughly 90 days, which includes for 3 growing seasons a year, excluding the winter months. During the growing season, students are tasked with weeding the field and watering the field in order to optimize the growing conditions of Stevia rebaudiana. Currently in Quadrant 1, it's a 17 treatment fertilizer study that looks to examine the effects of different fertilizers in order to optimize the growing conditions of Stevia rebaudiana. These include fertilizers such as cow manure, chicken manure, vermicompost, fungicide, and neem trees. Over here in Quadrant 2, we have the same 17 different growing treatments as well as different effects on harvesting frequency, which include 1 time a year, 2 times a year, and 3 times a year. In Quadrant 3, we have the same 17 treatments, but we're also looking at how growing density can affect the growing conditions of stevia. In each bed, they can include 21 plants, 35 plants, and 45 plants of Stevia rebaudiana. Now let's go pick up with CJ.
Hello everyone, my name is Clarence Hicks. I'm a second-year graduate student here at Fort Valley State University. And I work with Dr. Samuels and Dr. Biswas researching biotechnology and plant science. So today throughout some of our tour, I know you've seen a host of many different exotic plants and trees, and a lot of these have medicinal properties. One thing also to be aware of is that these plants do shed a lot of leaves, stems, and fruits that we can't all use personally. Now, waste management might be an issue in some areas, but here at Fort Valley State, we've come up with a pretty effective system to manage our excess. And I'll take you over to our composting pile here and show you a little bit more in detail what we're doing with that. So this is one of our composting setups here that we have. Now, you may know already, but composting is just simply a way of taking waste materials, mostly plant source, some animal source, and turning them right back into compost, food for the plants. So here at Fort Valley State, we've got a lot of plants that we work with in our greenhouse. So stevia being one of the more common ones. So after we work with our stevia and harvest it, we can process it and it turns into this nice brown component of compost. So with composting, you would want about a nice ratio of 4:1 carbon to nitrogen sources. Now with this ratio, you'll have enough food for the microbes to work down into plant food, and at the same time, you won't have a too hot or too acidic compost pile. Now, we've got several things here as a component of our brown compost. This here is a little help from the vet tech guys, if you know what I mean. And here is our stevia that we've grown in the field as well as the greenhouse, processed down. Now, taking a quick break from this, let me take you over to our intercropping fields so we can show you what we have going on there. And welcome to our intercropping field, where we're actually looking at a way of growing more plants in less space. You can see here we do have several raised beds. Right pictured here is some stevia. Some of them have some genes of interest that we're looking at. And all throughout here you see many rows of peaches. Just taking a little bit further down looking too. So as I stated earlier, one of the things we're looking at here is growing more plants in less space. Well, at the same time, there is a issue in the southeastern United States for fruit growers that we're looking at, and that's called PTSL, or peach tree short life syndrome. Now, we do have over 8 varieties here and over 1,200 peach trees that we are examining currently to see some of the root causes of this phenomenon and probably try to put a dent in that problem. So as you can see, some of our peaches are blooming pretty nicely here already, and some of these will bloom now, some will bloom a little bit later into the summer. But they do have a unique relationship with the plants here. And just taking a further look down into some of our lettuce beds here, I'll explain a little bit more about that. So as these peach trees grow, they will provide shade and some wind coverage to our lettuces and other crops that we do grow down here. And at the same time, when the leaves and fruits will fall down into the soil as well, they will eventually compost down its nutrients that the plants can use. As you can see here, we do have 3 lettuce varieties right here. There's some butter crunch lettuce, red leaf, as well as some romaine. And now, just taking a quick break from here, I'm gonna take you over to our other stevia field so we can see what Daryl's doing over there.
Hi everyone, my name is Daryl Sparks. I'm a second-year graduate student here at Fort Valley State University, and what you're looking at is the stevia field. Here, you have a stevia that you will notice is bigger than others. Why? Because it's actually from 2012. Most of the stevia that you see out here were planted, but they actually died, because what you don't know is stevia is a perennial crop that cannot survive cold temperatures. So when we noticed that this stevia was actually big and stevia like this, we took a piece of their stem and we mass produced it in our hydroponic system. And that's what actually led to the in vitro study of this great crop. If you come over here, you'll see lettuce that we grew and also turmeric. If you look right here, you'll see that we have romaine lettuce, red lettuce, and butter lettuce. We did a study on these 3 different types of lettuce to see how well they will grow traditional farming versus moderate farming. This is the first 5 weeks, and this is the second 5-week study. And as you can see, the first 5 weeks have matured way better than the second 5 weeks. If you come over here, you'll see our turmeric crop. Turmeric is a cash crop, and one of the reasons why is because it has medicinal uses, and that is why it is very high in demand. The medicinal and traditional uses of this crop intrigued me, so I decided to dedicate my master's on research to finding out more about this great crop.
So this is some of the experiments that we're going to be working on, which include plant tissue culture and chemical production. Oh, didn't see you there. Welcome to our lab. My research is on in vitro multiplication of selected Stevia rhodesiana. The aim of my research is to generate a series of protocols for the laboratory to further future research for incoming graduate students. In this experiment, I use biotechnology to multiply and to study the development of Stevia rhodesiana. Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known composition. It is widely used to produce clones of a plant in a method known as micropropagation.
Hi, today I'm working on my thesis project. The title of my thesis is called Investigating In Vitro Clonal Propagation of Various Tomb Germplasms Grown at FVSU and Testing the Fidelity of the In Vitro Raised Plants Using Molecular Markers. So basically, I'm going to harvest germplasms from different places throughout the Fort Valley State farm. I will bring the plants into the lab for the sterilization process. After properly sterilizing the rhizomes, I will then culture the plants and leave them for 4 weeks in the nursery room. After the 4-week period, I will then culture the egg plants again and put them on the root IAA and shoot BAP media to help with organogenesis.
First, the media packet is removed from the fridge and allowed to warm briefly. Next, the packet is opened with care to reduce the chances of introducing any foreign substances. Here, the students have pre-measured 1,000 milliliters of distilled autoclave water. 900 mils will be added to an autoclave flask, leaving behind 100 mils to rinse any remaining residue from the media packet. Next, 30 grams of sucrose is measured and added, and the mixture is stirred into a solution. Next, the media will be adjusted to a pH of 5.8, which is an optimal environment for the uptake of minor and major minerals. Following pHing, agar will be added to solidify the media. Next, Daryl will add autoclave tape and introduce the media to the autoclave. Following the autoclave process, the media will be taken to a sterile laminar flow head. Once properly cooled, any required hormones will be added at the appropriate concentration for their desired effect. Next, the fully prepared media will be aliquoted into individual culture vessels to receive their plants. Here, Jeffrey Lester is excising stevia cuttings to be used as explants. Jeff will start by sterilization of his equipment. His forceps and scalpels will be sterilized by dipping them in alcohol and placing them into the incinerator to heat sterilize, followed by a short cool down period. Jeffrey starts by cutting a section containing 2 sets of nodes, leaving the bottom inner node section slightly long. Any excess leaves will be removed, and the explant will be established in its culture vessel. Finally, the vessel is sealed with micropore tape that allows for gas exchange of oxygen and CO2, but will prevent contamination. After being sealed, the vessel will be properly labeled for storage in the culture room. Here, Daryl is culturing a plant, turmeric. However, given the differences between stevia and turmeric, the culturing process is slightly different. The explant in this case is a rhizome with an actively growing bud. Daryl will start by sterilization of his equipment. His forceps and scalpels will be dipped in alcohol and then placed into an incinerator to heat sterilize them, followed by a short cool down period. Daryl starts by trimming the rhizome, making it more uniform, removing the outer shell, and taking care not to harm the bud. The trimmed explant is then established in its culture vessel. Finally, the vessel is sealed with micropore tape to allow for gas exchange of oxygen and CO2, but will prevent contaminants from entering. Following sealing the vessel, the tube will be properly labeled for storage in the culture room.
So as you can see here in the plant tissue culture room, these are some of the various different plants that I have growing under controlled conditions to generate data from my research. we have plants listed at various media concentrations. Shoot and root induction fall under the category of organogenesis, the production of an organ system of a plant. Organogenesis is defined as the development of organs like roots, shoots, and flowers either directly from an explant or from the callus culture. The embryo is not considered an organ because the absence of a vascular system in its independent existence. Murashige and Skoog media and woody plant media were used in these plant tissue culture experiments. Plant media was augmented with various concentrations of plant growth regulators, or PGRs. Plant growth regulators such as BAP, or 6-aminobenzoylaminopurine, and NAA, or 1-naphthalenic acetic acid, were used in my shoot induction experiments. In my preliminary results, I found that media supplemented with 2.5 milligrams per liter of BAP was best at initiating shoot growth. Plant calluses, or calli, are a growing mass of unorganized plant parenchyma cells. In living plants, calluses cells are those cells that cover a plant's wounds. In biological research and biotechnology, callus formation is induced from plant tissue samples after surface sterilization and plating onto tissue culture medium, in vitro in a closed culture vessel such as a petri dish. The culture medium is supplemented with plant growth regulators such as auxin, cytokinin, and gibberellin to initiate callus formation or somatic embryogenesis. In my experiments, I am using MS media supplemented with different growth regulators to initiate callus growth in stevia levels. In my preliminary results, I have found that media supplemented with 2.0 milligrams per liter of 2,4-D supplemented with 1.0 milligrams per liter of BAP was the best at initiating callus formation in the leaves of the stevia plant.
So after deforming of the new plants, I will test the clonal fidelity of the new plants using molecular markers along with the PCR thermocycler technique. PCR stands for polymerase chain reaction. Upon completion, the clones will go through the process of getting exposed and acclimated to the outside to grow. I used rhizomes parts of the plants for the tumor sterilization. My main target when selecting rhizomes were the shoots. I targeted shoots that were at least 1 inch long to give the plants the best chance of survival.